So we will start, because we have discussed this 7 step. So now we will see the last one, which is important for our exam and conceptual also for understanding, phagocytosis. So next topic we are going to discuss is phagocytosis. So next topic we are going to discuss will be phagocytosis. Right. So in phagocytosis, first we have to understand what are the cells involved in phagocytosis, which cells are responsible for phagocytic functions, right? So we should understand what are the cells responsible for phagocytosis. We all know that some of the basic cells, like we all know neutrophil is a phagocytic cell. We all know monocyte and macrophage, they are also phagocytic cell. But very rarely we understand that this eosinophil is also a phagocytic cell. So just remember, eosinophil is also a weak phagocytic cell for parasites, right? So that is the important thing we have to remember. For parasites, eosinophil is also a weaker phagocytic cell. So number one, we have to understand what are the phagocytic cells. These are the phagocytic cells, right? Now we will see the second thing, what are the events in phagocytosis. That is also important to understand, what are the events or steps of phagocytosis. So now I am going to discuss steps of phagocytosis. What are the steps of phagocytosis? So there are 3 steps in phagocytosis: recognition and attachment, number 1; engulfment, number 2; and killing and degradation, number 3. So there are 3 steps. First they will recognize, then they will attach to the bacilli, number 1. Then they will engulf them, number 2. And after engulfment, they will kill them and then they will degrade their dead body, number 3. So these are 3 steps. So we will see one by one. So number 1 is called recognition and attachment. So recognition and attachment is the first step, and try to understand this statement is a very, very important and very conceptual statement, right? Recognition and attachment. So first step is recognition and attachment. It's a conceptual step, and how we are going to understand this, now we can see this, right? Suppose I am showing you this is the phagocytic cell. It can be any phagocytic cell. For example, I am showing you this is the macrophage, right? This is the macrophage, a phagocytic cell, and here I am showing you a bacteria, right? So what is the basic problem for this macrophage? This macrophage is not able to identify properly this bacteria for the phagocytosis, means recognition is not there. So how this macrophage is going to recognize this bacteria? How? For identification of this bacteria, for recognition of this bacteria, see the word I am using, for recognition of this bacteria. Bacteria will require some special substances on their surface, and these special substances, so I am just drawing here, these are the special substances which will coat the bacterial body, and they are called as opsonin. These are opsonin, right? So that is the point we have to keep in mind. These are opsonin. So opsonins are compounds which are required for recognition of the bacteria, and they help in phagocytosis. They enhance phagocytosis. So what are the opsonin? These are the substances. Remember, this was the question in All India exam. Opsonin are the substances which helps in recognition, and enhances phagocytosis, So that was the answer in All India exam. What are the opsonins? Opsonins will enhance phagocytosis. How they will enhance phagocytosis? They will coat the bacteria or any cell. Not only bacteria, they can coat any cell, RBC, WBC, platelet. So they will coat any cell, and because of the coating of the cell or microbe, they will be able to they will be able to identify by this macrophage or phagocytic cell. So that is why it is written recognition and enhances phagocytosis. So what are the substances which are considered as opsonin? Another All India question in 2017 and 18. This question had been asked, okay, what are the opsonins? So remember, what are the opsonins? So example of opsonin is antibody and complements. So these are the two things. Which antibody is the best? Answer will be IgG antibody is better than IgM antibody. So this is important. So IgG antibody is better than IgM antibody, right? IgG antibody is better than IgM antibody for opsonization. So this was the older All India question. IgG antibody is better than IgM, right? Amongst the complement, this was the previous year All India question. C3b is an opsonin. So this was the previous year All India question. Which complement is opsonin? C3b is opsonin, right? So C3b, immunoglobulin or IgG, IgM will be coating the bacterial cell wall or bacteria, and this will be opsonizing, right? So this is called opsonization. So this is called opsonization, and this opsonization is essential for recognition, right? So once they had been recognized, so opsonin is for recognition. So after recognition, what will be the next step? Now the question is after recognition, right? So now you can see they had been recognized because of the opsonin. So what will be the next step? So after opsonization, I'm going to show that this is the macrophage, right? This is the macrophage. Macrophage, they will be having scavenger receptors on their surface. They will be having scavenger receptor. So what I'm showing you here, this is the scavenger receptor. I'm showing you here, this is called scavenger receptor, right? The scavenger receptor is present on phagocytic cell. It can be macrophage also, eosinophil also, neutrophil also, right? So this scavenger receptor will combine with the opsonin, right? So opsonin, suppose this is the opsonin. This is the opsonin which is coating the bacteria, right? And this was the bacteria. This was the bacteria inside, right? So now we can see bacteria was coated with opsonin. So opsonin is combining with the scavenger receptor. So I am taking example of this opsonin here is the IgG, right? So this is how it will be combining. So this is called attachment. This is called attachment. So now we can see that is why I wanted to tell you, okay, this is the important concept which was questioned in AIMS exam also. Okay, after opsonization by immunoglobulin C3b or this thing, what will be the next step? So after opsonization, will they form pseudopod or will they be having attachment of a scavenger receptor to the immunoglobulin, right? So answer was attachment of the scavenger receptor to the immunoglobulin, not the pseudopod formation, right? So that is why I started like this. It's a good concept, right? First recognition, then attachment, and for attachment what they are going to use is scavenger receptor, right? So for attachment they are going to use scavenger receptor with IgG. So here I will tell you what was that exact language in AIMS exam, because in exam scavenger receptor, there is a family of scavenger receptor which is given in exam, Fc gamma 3 receptor. So these are the scavenger receptor examples. So these are the example of the scavenger receptor. So Fc gamma 3 receptor will combine with Fc fragment of immunoglobulin, right? Fc fragment of immunoglobulin, or it may combine with the complement C3b. So this is the step of attachment, right? So this is called attachment. Fc gamma 3 receptor, which is a scavenger receptor, will combine with Fc fragment of immunoglobulin IgG or IgM, or it will combine with complement C3b, right? So now we can understand this scavenger receptor is combining with this, and this is called attachment. So recognition and attachment, this is the first step. After that what will happen, right? So second step, as I was telling you, second step for phagocytosis is the engulfment. Second step for phagocytosis is the engulfment, right? So as I have told you, when bacteria will attach to the scavenger receptor of the macrophage, see, when this bacteria is attached to the scavenger receptor of the macrophage, right? Scavenger receptor of the macrophage. So what will happen inside the macrophage? Inside the macrophage, there will be increase in intracellular calcium concentration, right? Because of this attachment, intracellular calcium content will be increased. Because of intracellular calcium content increase, this will activate extra amount of guanosine triphosphate. So GTP will be formed with the help of intracellular calcium signals, and this GTP will cause polymerization of actin filament. Polymerization of actin filament. We know that what is actin? A contractile protein, right? So there is a polymerization of actin. So when there is a polymerization of actin, what will happen? Contraction. So what will happen when there is a contraction? Who will contract? The cytoplasm will contract. And now because of the contraction, there will be foot-like process which will be coming out from the cell. So these are foot-like pseudopods. So these are called as pseudopod formation. So now we can see this is how pseudopod formation will start. So because of the polymerization of actin, you are seeing pseudopod formation, right? So pseudopod is formed. Pseudopod formation. Right. So as I said, because of the intracellular calcium increase, signal will be generated, GTP will be produced, which will help in polymerization of the actin. Because of polymerization of the actin, foot-like process will be formed, which will start moving towards the bacteria for the next step, which is called as engulfment. Right. So now after this pseudopod formation, what is going to happen? What is going to happen now? We will see that. Right. Pseudopod has been formed. And now this macrophage pseudopod will engulf this bacteria. So this bacteria is inside the vacuolar region. This is called the phagosome. So now it is phagosome. So you can see that pseudopod has engulfed them. So this is called engulfment. Now engulfment has been happened, and this structure is called as phagosome. This is called as phagosome. So inside the phagosome now bacteria has been engulfed. Later on, this phagosome will combine with another structure which is called as lysosome. Right? So lysosome will attach to this bacteria containing phagosome. So this entire structure which is containing lysosome and phagosome is called as phagolysosome. So now phagolysosomes are formed. So up to this is called step of engulfment. So bacteria has been engulfed by the pseudopod formation, and after the engulfment, the phagosome fuse with the lysosome and phagolysosome will be formed. What is the purpose of phagolysosome? Purpose of phagolysosome is to kill the bacteria or kill the microbe within this phagolysosome, and that is how third step will be evolving, right? So now we will see the third step. Third step in phagocytosis is killing, right, and degradation. Killing and degradation. So third step is killing and degradation. So what happens in killing and degradation? As you can see, I have shown you phagolysosomes, right? So again I will draw that diagram so that we can understand more clearly. This is the macrophage. This is the lysosome. As I have told you, lysosome will be connected to the phagosome, and phagosome will be having bacteria, right? Lysosomes, they will be having lysosomal granules, right? So now I am showing you these are lysosomal granules, right? So lysosomal granules, when they are exposed to the bacteria, this lysosomal granule will be having rupture, right? So as I said, whenever these lysosomal granules will be exposed to the bacteria, this granule will rupture. So this rupture of lysosomal granule is called as respiratory burst. Remember, this rupture of the lysosomal granule is also known as respiratory burst. So you might be reading all these languages in your textbook, and that time you may not be understanding what is respiratory burst. Rupture of the lysosomal granule when they are exposed to the bacteria is called as respiratory burst. right? So after respiratory burst, what will happen? After respiratory burst, what will happen? There will be release of an enzyme which is known as NADPH oxidase from the granule, right? So NADPH oxidase will be released from the granule, right? So NADPH oxidase is also called as respiratory burst oxidase, right? So that is why it is also called as respiratory burst oxidase, also known as phagocytic oxidase. Phagocytic oxidase. So these are the various names for NADPH oxidase. Phagocytic oxidase, respiratory burst oxidase. Why I wanted to write phagocytic oxidase? Because initial two alphabets of the phagocytic oxidase is the gene which controls function of the NADPH oxidase. So what I am telling here, try to understand what I am telling here. There is a gene called as FOX gene. FOX gene. And this FOX gene controls the function of NADPH oxidase, right? So phagocytic oxidase gene. So this is very important to understand. Phagocytic oxidase gene controls the function of NADPH oxidase. So once again I repeat, when phagolysosome is formed, lysosomal granule will be exposed to the bacteria and then they will rupture. After rupture, NADPH oxidase will be released, and what will happen next? When NADPH oxidase will be released, they will produce free radicals, right? So NADPH oxidase will produce free radicals, and this free radicals will kill the bacteria. So this will be involved in killing. So that is how bacteria will be killed within the phagolysosome. So NADPH oxidase, now you can understand what is the function of NADPH oxidase? To form the free radical, and that free radical will be killing this bacteria. So now the dead body of bacteria is still there. So how it will be removed? By degradation, right? So lysosome will be having one more enzyme. So after killing, see this, what I am writing, after killing, there will be lysosomal enzyme, another lysosomal enzyme which is called as acid hydrolases. Acid hydrolases, right? So there is a acid hydrolase which is present in the lysosomes, and this will cause degradation of bacilli, right? So dead body of bacilli will be degraded by acid hydrolases present in the lysosomes. So this is how killing and degradation will be completed, right? So that is what I wanted to explain you. Three steps of the phagocytosis: recognition and attachment, recognition by opsonin, attachment with scavenger receptor, then engulfment with the help of pseudopod, and after that phagolysosome formation, which will help in killing and degradation. Killing is done by free radical produced by NADPH oxidase, and degradation of the dead body of the bacilli will be done by the acid hydrolases. So these are the important points we have to remember for phagocytosis. Here I want to discuss this free radical production and phagocytic killing. So there is another important step which we have to understand is the phagocytic killing. This topic is again a subtopic of the phagocytosis, phagocytic killing. In phagocytic killing, as I have told you, free radical, I had given example, but what we have to understand, phagocytic killing can be both oxygen dependent and oxygen independent. So in other words, what I wanted to tell you, phagocytic killing can be oxygen dependent, right? Oxygen dependent means free radical mediated, right? Oxygen dependent means free radical mediated. This is the number one. So phagocytic killing is oxygen dependent via free radical, and it may be oxygen independent without using any free radical. So these are two methods. These are the two methods of phagocytic killing, right? So I am writing here phagocytic killing by two methods. Free radical method is called oxygen dependent, and without free radical it is oxygen independent. Which is the most effective method? That is how examiner asks. Most effective method is the oxygen dependent method. So here we have to understand oxygen dependent is the most effective method. right? So oxygen dependent is the most effective method. So oxygen independent mechanism, what are the substances involved? In oxygen independent mechanism, there is a involvement of, there will be involvement of lactoferrin, there will be involvement of lactoferrin, lysozyme, lactoferrin, lysozyme, and third one is the important one, that is called major basic proteins. Major basic proteins, and this was the question in all India exam. Okay, major basic proteins are present in granules of which cell? Question is, major basic proteins are present in granules of which cell? So here we have to remember that major basic proteins are present in eosinophilic or eosinophil cytoplasm, right? So they are present in eosinophil cytoplasm, and that was the answer in exam. So major basic protein is present in eosinophilic cytoplasm. So this way also we can connect कि eosinophil is also a phagocytic cell where they will kill the bacteria or parasite which is oxygen independent mechanism by using their protein or granular protein that is known as major basic protein, right? So these are the three things: lactoferrin, lysozyme, major basic protein, which are oxygen independent. So come to the basic one. The important one is the oxygen dependent killing. So we were talking about oxygen dependent, the most effective method. How this most effective method will be performed? This will be performed through the help of NADPH oxidases. As I have told you, phagocytic oxidase, NADPH oxidase. What they are going to do? NADPH oxidase in our cell, NADPH oxidase will convert oxygen into superoxide free radical. See this, what they are going to do? They are going to convert oxygen into superoxide free radical, right? So oxygen into superoxide free radical. So this is the superoxide free radical. So this superoxide free radical is formed by NADPH oxidase. Now this superoxide free radical will be degraded into, this will be degraded into hydrogen peroxide with the help of one enzyme which is called as superoxide dismutase. So superoxide dismutase will convert superoxide into hydrogen peroxide. Hydrogen peroxide is also a free radical. It's also a free radical, but remember, this is the weaker agent for killing the bacteria, right? So hydrogen peroxide is also a free radical, but it's a weak one. So still we are not seeing the most effective bactericidal agent in phagocytosis. So what will be most effective bactericidal agent? So what will be most effective bactericidal agent? When they will have involvement of myeloperoxidase along with halide ion or chloride ion. So myeloperoxidase and halide ion. So in presence of myeloperoxidase and halide ion, this hydrogen peroxide will be forming hypochlorite ion, right? So they will be forming hypochlorite ion. So in presence of myeloperoxidase and chloride, they are forming hypochlorite. They are forming hypochlorite, right? So these hypochlorites are most effective bactericidal agent, right? So most effective bactericidal agent is the hypochlorite, and that is the important MCQ which examiner ask in exam, right? So hypochlorite is the most effective bactericidal agent within the phagocytic cell. So sometime examiner will be asking hypochlorite, sometime examiner will not be writing hypochlorite, they will be simply writing free radical, and sometime examiner will be writing H2O2 MPO halide system. So this is also called as H2O2 MPO halide system, right? So this was the question which had been asked in All India NAIMS exam, right? So this is how this language had been used, H2O2 MPO halide system, right? So just keep that in your mind, this H2O2 MPO halide system is the most effective bactericidal agent within a phagocytic cells, right? So this is how we can see and we can understand ki why I started like this, right? Ki phagocytic killing is oxygen dependent and oxygen Independent. Dependent is the most effective method where NADPH oxidase converts oxygen into superoxide. Superoxide will be again converted into hydrogen peroxide. Hydrogen peroxide in presence of MPO and halide, they will be forming hypochlorite, and this hypochlorite will be the most effective method to kill the bacteria within a phagocytic cell. So this is also called as H2O2 MPO halide system, or also known as free radical mediated method of killing of the bacilli or microbe within the phagocytic cells. So this is the important point about phagocytosis. Now we will see what is the clinico-pathological correlation of this phagocytosis. In clinico-pathological correlation, see what I am telling here is what I want to tell you here about the clinico-pathological correlation. Clinico-pathological correlation. Clinico-pathological correlation of phagocytosis is with a disorder known as chronic granulomatous disease. So this is associated with one example of chronic granulomatous disease. So here I am writing full form is chronic granulomatous disease. Chronic granulomatous disease is an hereditary disorder which will be having autosomal recessive as well as X-linked recessive, both pattern of inheritance, which I will discuss later on. Right? So as I said, these are the hereditary disorders, right? In this hereditary disorder, what is the basic defect? What is the basic defect? Basic defect is the respiratory burst. So in this, what you are going to see that respiratory burst is defective. You remember what is respiratory burst? Just now I have told you. Respiratory burst means rupture of the lysosomal granule. So this rupture of the lysosomal granule will be defective. So this is the basic defect in the chronic granulomatous disease. Respiratory burst will be defective. Why this respiratory burst is defective? Why this respiratory burst is defective? This defect is because of the mutation. See, this defect is because of the mutations, right? Mutations in gene. So which gene mutation? So this respiratory burst is defective. Lysosomal granule will not rupture. Why there is no rupture? Because of the mutation. Mutation of what? There are genes which are controlling. If you remember when we were discussing about FOX, NADPH oxidase, that time I told you NADPH oxidase is controlled by a gene called FOX gene, right? So now we can see there is a mutation in FOX gene. Mutation in FOX gene. And FOX gene, there are two locations. They can be membranous. So FOX gene can be membranous, which is present on the membrane. So membranous FOX gene mutation, or it can be cytoplasmic FOX gene mutation. So membranous or cytoplasmic. So NADPH oxidase is not released because of the respiratory burst defect. NADPH oxidase is not released. Why NADPH oxidase is not released? Because of the mutation in the FOX gene. What is location of the FOX gene? It's membranous. It can be cytoplasmic, right? When they are involving membranous, this will be called as membranous type of chronic granulomatous disease. When they are involving cytoplasmic mutation, this will be called as cytoplasmic chronic granulomatous disease, which is most common. So remember, membranous is the most common type. So this point was the question in All India exam. Most common type is the membranous one, and membranous will be X-linked recessive. That is why I wanted to tell, right? Okay, this will be X-linked recessive disorder. Cytoplasmic will be less commoner, and they will be autosomal recessive. X-linked recessive was All India question in 2016. Membranous type of chronic granulomatous disease is the X-linked recessive. Or when examiner simply ask, as it was asked in All India exam, ki overall chronic granulomatous disease, which pattern of inheritance is most common? X-linked recessive pattern of inheritance will be most common, right? So now we can see that mutation is in FOX gene, membranous and cytoplasmic. Membranous is X-linked recessive, more common. Cytoplasmic is autosomal recessive, less common. And what are the genes responsible for this one? So again, I will tell you, this was the question in All India exam. When we are talking about membranous, it is glycoprotein 91 FOX gene mutation. So that was the answer, right? So GP91 FOX gene mutation. GP91 FOX gene mutation is associated with membranous type of chronic granulomatous disease, whereas autosomal recessive is associated with two proteins called as P47 and P67 FOX gene mutation. So 47 and 67 FOX gene mutation is responsible for autosomal recessive type, right? So autosomal recessive is 47 and 67, whereas when we are talking about membranous, the most common type will be GP91 FOX gene mutation. So because of this mutation, what is happening? Respiratory burst is defective. Because of defect in respiratory burst, NADPH oxidase will not be formed. So bacilli will be not killed. Which type of bacilli? Catalase positive or catalase negative or both? So this question we have to understand. This was the question in DNB exam previous year and all India exam also. This patient, because of this mutation, this patient will be having history of recurrent infection with catalase positive organism, right? Catalase positive organism example which has given in DNB exam was Candida and Staphylococci, right? So recurrent infection by which type of organism? Catalase positive type of organism, and their examples are Candida and Staphylococci, right? So they will be having recurrent infection with catalase positive. Why catalase positive? Because when catalase positive organism is there, so we all know that catalase will degrade hydrogen peroxide, right? So they will degrade hydrogen peroxide. So if hydrogen peroxide is degraded, so now you can understand when we were seeing that oxygen dependent mechanism, hydrogen peroxide, then MPO halide will be forming hypochlorite ion. So when this hydrogen peroxide has been degraded, so that halide will not be formed, and that is why recurrent infection by catalase positive organism will be seen in this chronic granulomatous disease, right? So this is the clinicopathological correlation of the phagocytosis. Phagocytosis is closely associated with this chronic granulomatous disease. Because of the phagocytic defect, they will be having recurrent infection, they will be having granuloma formation, that is why the name is chronic granulomatous disease, and this is example of the primary immune deficiency disorder. So primary immunodeficiency will be present in this patient. So remember this point, these are the example of the primary immunodeficiency disorder. So they will be having primary immunodeficiency.